some new participants this evening in Discoveries in the Making, and I'm just thrilled. So my name is Lori McMahon, and I'm the Dean of the Graduate School at UAB, and I'm thrilled to have all of you here this evening. And so for those of you who haven't tuned in before, I'll just share a little bit about the goal of Discoveries in the Making. So my background is neuroscience, and I run a basic science research lab. And over my career, I've been really passionate about our responsibility as scientists to share with a non-expert audience. And I've talked a lot about that with my own students in my laboratory. And since I've been Dean of the Graduate School for the past 5 years, I've been really passionate about extending that to other students and making sure that they know their responsibility. We get our funding from federal resources, from state resources, sometimes it's even from donors. And so I strongly feel that it's our responsibility to share back to all of those taxpayers, all of those donors who provide funding for us to make the discoveries that we do. So our students and our faculty, we all get very comfortable with speaking to others in our discipline. We know the lingo, we know the assumptions, we know how to talk to our peers. But often what we're not that great at doing is talking to a non-expert audience. And so there's no way for us to really share our discoveries with others who aren't in our field if we can't communicate and to use words that all can understand. And so it's out of this passion of sharing our discoveries with others that Discoveries in the Making was developed. And so we've been doing this now for about 3.5 years. And so before COVID times, we were doing this in a local science-inspired bar called the Lumbar. And so the Lumbar is set up for this kind of activity. It's a science-inspired venue. And so we had been going to the Lumbar to share our discoveries. And so since COVID, Kim Eaton, who's our Director of Communications in the Graduate School, has continued to help us share our discoveries through this virtual Zoom activity. So we are here for you. Our speaker, Sabrina Heiser, is here for you. She wants to share with you the discoveries that she is making. And so we really want to have a conversation. We want questions. Sabrina wants you to ask questions. So you can do that by putting your question in the chat. You can also do that by raising your hand. I think, you know, when we are doing this in person, we really encourage questions during the presentation. It's a little bit harder to do that with Zoom. So we'll have Sabrina present her work to you, and then we'll take questions at the end. And we're not on any particular time frame, so we'll keep the conversation going as long as you have questions. And so I just encourage you to take advantage of this time that we have this evening and really talk to Sabrina about her work. So Sabrina, I'm going to turn this over to you so that you can share with us what you're doing.
All right, thank you. Just gonna start sharing the screen. All right, is this working?
Yes, looks great.
All right. Well, thank you very much for this introduction. And I would also like to really thank the graduate school, especially Dean McMahon and Kim Eaton, for organizing this and keeping it going, moving it virtual, to provide me with this opportunity to present part of my research. And today I would like to tell you a little story about the seaweed forests in Antarctica. So in order to tell you why we should care about seaweed forests, I'm going to back up a little bit and start with an example that we might be a little bit more familiar with. So this is a picture of a rainforest, which you either may have visited yourself or you've seen in documentaries, or maybe even in your biology book when it started talking about biodiversity. So in this image, we can see a variety of different plants. We can see a lot of trees and bushes, so plants different sizes. And what we can see that within those plants and trees, we have a multitude of different insects that are supported by them. We've got bird species that live amongst them and forage amongst them. We've got other invertebrates and vertebrate species that benefit from these plants. And what I'm talking about here, the biodiversity, is talking about diversity in terms of having a lot of different species. However, the diversity that I'm going to talk about tonight is a little bit different because I'm going to talk about the diversity that we see within species. So just like you and me having different eye or different hair color. But why does diversity within species actually matter? So the example that I like to use here is geared towards Birmingham, because we are here in a city that has a long history of iron and steel production. So it's tightly connected to the Industrial Revolution. And the example that I like to use is the peppered moth that you can see in this image here. Is sitting on the tree. And we have two different color morphs of those peppered moths. So the light colored one, which I've circled on the top here, and the dark colored peppered moth, which is circled on the bottom. And they like to sit on birch trees. And as you can see, the bark of birch trees is generally light colored. So when a predator comes along, like the bird over here, it is able to see those light-colored moths a lot better, the dark-colored moth a lot better than the light-colored moth, and is able to pick the dark ones off the trees. However, as the Industrial Revolution happened, we had a lot of soot that was produced by the furnaces and things like that, which would then come and they would cover the bark of those trees, which all of a sudden means that our dark-colored moths are able to blend in with their background a lot better than those light-colored moths. And the birds were starting to pick up the light-colored moth because those are the ones that they are able to see a lot better. And without that initial diversity that we saw within the moth, this species may have not survived, because all of them would have been eaten. So this is in general why diversity matters. It allows species to adapt to changes that are happening in the environment, either naturally or induced by something that we are doing. So with that in mind, I would like to take you all with me to Palmer Station or to Antarctica for the next 10 minutes or so to talk about why diversity within species matters there just as much. So I find that Antarctica generally seems a little bit neglected at the bottom of our maps. It's usually just a small little strip at the bottom. But if you look at the map and compare it to the size of the US, we actually find that it's as big as the US and Mexico combined. So it's pretty large. And the research that I'm going to be talking about takes place at the Western Antarctic Peninsula, which is down here on the map up here. It's over here, so that's just south of South America. And for the rest of the talk, I'm just going to refer to it as the peninsula. And what makes this area so interesting is that we have a really high marine biodiversity there. So we have a lot of different organisms that are living there, but at the same time, it is also one of the fastest warming places in Antarctica. So it's already seen a lot of change, but there's also a lot more change that is still going to happen in the future. And at Palmer Station, which is the American research station on the peninsula that the research that I'm going to be talking about is taking place, at Palmer, we still have enough sunlight that we get these really dense and lush macroalga forests or seaweed forests. So I'm going to be using both words, seaweeds and macroalga, interchangeably. And this is what those macroalga forests look like. So on the top left here, we have a big brown sort of canopy forming seaweed. The light spots on it that you can probably just about make out. They're actually little gas-filled floats, which is why this seaweed is able to stand upright. And this one is about 6 feet tall, but they can grow even higher. And then on the right here, we have another brown seaweed that doesn't have those gas-filled floats, so it's just draping over the seafloor. And those can get about 20 feet long commonly, but they can get even twice that long, so that they're fairly big. And then underneath those big canopy-forming seaweeds, we have red seaweeds that covers the other available space. So either in between the seaweeds, or they're even growing underneath the ones that are draping over the seafloor. And just to give you an idea of the size of those, the one on the bottom right is about the size of both of my hands together. And the biomass of those seaweed forests is actually equivalent to the biomass of the giant kelp forest that you can find in California. So that might be something that you've either seen yourself or in the aquarium there, or in documentaries. And this means that those seaweed forests provide a lot of structure and homes for organisms to live in, but they are also a potential food source. So just like the trees and plants that we find in the rainforest that I showed you earlier. So one kind of organism that is living in those seaweed forests are these little shrimp-like organisms that are also called amphipods. And they're about the size of the fingernail of my little pinky. So they're really abundant. They're in such high numbers that people often like to call them the insects of the ocean. And just to give you an idea of how many we find, so within a meter square of bottom area, within the brown seaweeds, we can sometimes find over 100,000 of those. So sometimes when you drop into the water and you get down to the bottom into the seaweeds, you can actually see them zooming around. It's pretty cool. And yeah, just imagine if you had a bush in your front yard that's the size of one, or that covers a 1 meter square bottom area, and there were 100,000 insects in there. It's quite crazy. There's quite a lot of them. And the organism that I'm going to tell you a little bit more about today is Plocamium cartilagineum, which is a red seaweed. And for the rest of the talk, I'm just going to call it Plocamium because Plocamium cartilagineum is quite a mouthful. And besides it being, I think, a really pretty seaweed, if you look at the intrinsic branching pattern, which actually amphipods can use quite well to hide in and hold on to, there are a number of other things that make it interesting. So first of all, Plocamium is very common. So not just around Palmer Station, but along the entire peninsula, wherever we are able to find seaweeds, we most often also find Plocamium. And then wherever we find it, it's also quite abundant. So there's quite a lot of it there. And in terms of the number of amphipods that they support, within a meter square, we are able to find over just over 10,000 of these little amphipods that live within the seaweed. However, most of the ones that live in it don't actually like to eat it. So why is that the case? So this is usually the point where I like to ask the audience and ask them to raise their hands about who knows someone who absolutely hates Brussels sprouts. And that is usually most people know someone or even hate Brussels sprouts themselves. And that is because those people are able to taste nasty chemicals that are produced by the Brussels sprouts in order to defend themselves against grazers, or I guess some human beings, because they are not able to run away as other organisms may be able to do. So they had to evolve different kinds of defenses. And this is also true for Plocamium and the amphipods. And Plocamium actually produces so many nasty chemicals that hardly anything likes to eat it. So that means that it is a pretty acquired taste. So just to recap, as I mentioned before, Plocamium is really common, it's really abundant, it supports a lot of amphipods, but hardly any of those will eat it. And as the group that I'm joined started looking more into this, and the group being the Amsler and the McClintock labs, which are both based in the biology department here at UAB, and then also the Baker lab, which is based at the University of South Florida, they started to look more into what kinds of chemicals they were producing. And what they found was that different individuals of Plocamium were producing different defensive chemicals. So for the rest of the talk, I'm going to visualize this by using different colors. So even though they actually all look the same, I like to visualize things because it makes it easier. And remember from earlier, diversity is good. We want diversity because it usually is good for a species. But the question is, how is this diversity distributed geographically? So what they did is they did surveys around the islands that are located around Palmer Station in order to see which individuals in which location were producing what chemical defenses. And what they found was that there seemed to be quite a strong geographic pattern. So certain individuals at certain sites were producing certain chemicals. And that indicates that there could potentially be an underlying environmental factor that is driving the distribution of those chemical defenses that we are seeing, meaning that maybe one side has got more predators present, maybe one side has got more nutrients available, or maybe the turbidity is different between the sites, meaning that if the turbidity is higher and there's less sunlight available, then the individuals may have less energy to produce certain chemicals. And then the next thing that they looked at was some of the genetics of those individuals. So they used a gene that is generally used to tell species apart, but you can also look at a little bit of genetic diversity within species. And they found two different genetic groups. So those were genetically different from each other, but not different enough to be different species. But the interesting thing here is that the individuals producing different chemicals sorted themselves quite well into those two genetic groups, meaning that there could be an underlying genetic difference between those individuals that it is explaining why different individuals are producing different chemical defenses. And this is kind of where I joined the project. So I'm really interested in trying to figure out what is driving this diversity that we are seeing. What are the underlying driving factors? So is it the environment, or is it the genetics, or maybe a mixture of the two? So in order to kind of look at that question, I got to go down for 2 field seasons, do a lot of scuba diving, which was pretty awesome, pretty cool. We conducted a lot of surveys and also a lot of experiments. And one image that I would like to point out is the one at the top right here, which was actually taken at World Women's Day. And we got an all-women's team to go out on the boat and go diving. And there's a fourth amazing female that was taking the picture at the time. So that was a pretty special day amongst a lot of other really special days. All right, back to the science. So the first experiments that we did in order to figure out whether the environment is impacting what chemical defenses individuals are producing were so-called transplant experiments. So just like you can transplant plants in your backyard, you can do that with seaweeds, a little bit more involved, but we did it. So what we did was we collected individuals from certain sites, and then we took them and transplanted them to a different site. And in order to do that, we used these concrete substrates that we strung them to, and then we left them out there for a year. So what we would expect is that when we come back after a year and collect them again, and if the environment is impacting what chemicals they are producing, any new tissue that grew would be producing a different chemical, or the chemical that was previously dominant at that site. And I can tell you it was quite a stressful experience, because we put these concrete blocks down there and string the experiments onto them, and then we leave for a year. And during this year, a lot can happen. Especially in Antarctica, we get a lot of icebergs that like to scrape across the ocean floor, and they like to wipe out anything that gets into their way. So when we went back and we found all of the transplant experiments again, there may have been some socially distant hugging underwater, or I guess a safe hugging underwater when we found all of the transplant experiments again. And as you can see, we didn't just find them again, but we also had some fresh growth on our transplant experiments. And from the some of the preliminary data that we've been able to look at so far, I can tell you that the chemistry did not change much in that new tissue that we collected from those experiments, meaning that it's likely not the environment that is actually impacting the chemical defenses that are being produced. Which brings me to the next part of my project, where I am trying to look at whether there are any underlying genetic patterns that could be correlated with the distribution of those chemical defenses that we are seeing. So instead of using the markers that were used before, I'm trying to develop markers that can look at this a little bit more closely. So I'm using tools very similar to 23andMe or Heritage DNA, any of those platforms where you can send a sample of your DNA to, and that is then analyzing and looking at where your ancestry came from. I'm using very similar tools to that in order to see how closely related individuals are to each other. So as I mentioned before, the seaweeds are sessile. So generally they can't travel very far. The only time they might be able to travel somewhere is if they get dislodged by an iceberg or during a storm, and then potentially reattach unless they float out to the open ocean or into deeper waters. They also are able to travel a little bit when they reproduce. So then when they produce spores or gametes, they're really tiny, so they likely don't travel very far. But overall, we don't really know very much about this. But that means that generally we would expect that those individuals that are closer to each other are more closely related to each other than those individuals that are further apart from each other. And then I'm trying to see how that genetic pattern compares to the pattern of the chemical defenses that we are seeing. So this is still work in progress. I'm still developing my markers at the moment, and obviously that has gotten a little bit delayed. And then I have about 700 or 800 samples that I have to go through and analyze. But what I can do is show you again some of the preliminary results. So the patterns that we're seeing so far are that individuals that are further apart from each other, that are producing the same chemistry. So remember that the same color means that they're producing the same chemistry. They are more closely related to each other than individuals that are closer to each other but are producing different chemistry. So this was really exciting when I started seeing those patterns, because it means that there seems to be an underlying genetic driver that can explain the distribution of the chemical defenses that we're seeing. So I'm excited to see how this continues as I analyze more samples. So overall, what I would like you to take away from this is that there are forests in Antarctica. So even though mostly when you see pictures from there, it's of icebergs and penguins, which are pretty awesome, but there are also seaweed forests there underneath the surface of the ocean. I would like you to take away that seaweeds are important. So they provide homes and food sources to other organisms, even if they are an acquired taste. So they're really important to the ecosystem and to the biodiversity. But also diversity within species is really important. And we are still in the need to understand what the drivers of this, that are maintaining this diversity is. As diversity is important for species to adapt to any changes that are happening. So with that, I would like to acknowledge the field teams, which have been absolutely instrumental to collecting the samples and the data, of course, and conducting the experiments in the field. I would like to point out Andrew Schilling, who was a PhD student at the time, who's actually the one who's doing all of the chemistry side of the project. Of course, my committee members and NSF, our funding source. And the last acknowledgement slides is very special to me because usually during these talks that I give at conferences or the last discoveries at the making talk, I'm not able to thank the Antarctic Support Contract or the people working for the Antarctic Support Contract directly, but because we've moved online, they're actually able to watch from Palmer Station in Antarctica. And I would like to thank them because without them, this work would not be possible. We wouldn't be able to do our research. I mean, they keep our lights on, they keep the buildings warm, hot water going so we can have a shower after diving. They keep our tanks filled for scuba diving. They keep our boats running so we can go out to the dive sites. They keep the labs running, provide us with mostly anything that we need. And if it's not on station, then someone will figure out how to make it for you, which is absolutely amazing. And then of course, the chefs that keep us really well fed, and everyone there is amazing and instrumental to this. So thank you. And with that, I'm happy to take any questions.
That was fantastic, Sabrina. Thank you so much for sharing that with us. And I'm so pleased too that we have all of these individuals who can tune in, including all of those from the Palmer Station. So thank you for taking your time to do that. And I also see that we have visitors from New Zealand and from Scotland, from Arkansas and from Washington State. So welcome and thank you for tuning in. So now is time for all of you to ask questions. And so you can either just unmute yourself and ask Sabrina directly, or you can put it in the chat. So, and Sabrina, I'll just start. So how genetically similar or diverse are these species that you have identified?
Yeah, so it's actually all the same species. So we have this.
Okay, so it is all the same species.
Mm-hmm.
Okay.
Yeah, we have this really high diversity within species, and there is not that much known yet about the general genetic diversity of seaweeds in Antarctica. There are some studies that suggest that the diversity isn't overly high, but.
So, but in your genetic analysis that you've done so far, how diverse, I mean, are you thinking that these are just mutations maybe? Could it be mutations in a couple of proteins that change the chemicals, and so how it is perceived by these animals as far as their taste? Could it be as simple as a mutation, or is it something different than that?
Yeah, that's a really good question. And I think some of that is probably better answered by looking at the chemical structures, which Andrew, who I know is in the audience as well, could probably answer a lot better. The thing that I do know is that some of the chemicals are very different. So some of them are cyclic and some of them are linear. So those are, as far as I understand, pretty major differences. That makes me think that it's not just one mutation that happened, yeah. But there's a lot that went on.
Great, thanks. Other questions for Sabrina? Don't be shy.
Hey Sabrina, it's Hannah at Palmer.
Hello.
I have two quick questions. So first, how do you, you know, that you said they were all the same species, but like, how do you distinguish between different areas? Like, what's your method for keeping that all straight? Like, do you call them subspecies, or like how do you distinguish between them?
Yeah, so we actually call them haplotypes, which basically describes different, a group that has the same sort of genetic sequence within a species. That's called a haplotype, if that answers your question.
Yeah, awesome, thanks. And then my second question was, how many sample sites do you have, or did you have, and do you see any continuity between like places that are shallower and maybe more surgy, even if they're distantly far apart, or yeah, just what sort of correlation do you see so far geographically?
Yeah, so that's a really good question, because that's something that we started thinking about, especially when we were down there as well. We actually have one site, so that's Nossel Point, where we have a very steep or a relatively steep wall compared to the other sites, and we also get a lot more currents that are coming through. And what we found there was that we had a very strong depth distribution into different kinds of chemical defenses, which made us think maybe there is a depth component to it as well. And we included that in the transplant experiments, as opposed to other sites where we had a less steep bottom, where we seem to have a lot more general distribution of the chemo group. So by that I mean there wasn't this stark shallow and deep difference that we saw at Nossel Point. And I honestly can't tell you right now how many different sites we went to in total. I think we have about 7 sites where we did complete transects. So for the genetics part of the experiment, we actually sampled at very set depth intervals. So once I've analyzed all of that data, I'm actually able to tell you more about that.
How fast do these seaweed grow?
That's another good question. They don't grow all that fast. So they generally grow for a couple of years. And I'm trying to remember, because we do actually have growth rate data, because we're trying, one of the other things that we were looking at was whether there's maybe a difference between, if you're producing a certain chemical that's really expensive to make, maybe you're growing slower. So I haven't quite looked at that data, but it's not a whole lot. So maybe, I don't want to make anything up.
And is it possible to grow them in a laboratory setting if you
Yes.
keep the salinity?
Yeah, so we actually have, at Palmer Station, we're really fortunate with the aquarium setup, because they have got flow-through tanks. So we're just getting the seawater from the outside, and that's how we were able to run experiments and keep them alive. And I did try to bring some spores back that I've started growing in the incubator here, but they do require some care.
Yeah. I could see an advantage to being able to recapitulate that in a lab setting. So then you could modify the environment, or change the environment to see if you can change the chemicals that they are releasing. And I mean, I know you didn't see a difference when you changed the environment, but there's still maybe something hidden there that you might be able to understand in a more controlled setting.
Yeah, no, that would be great. I think the difficulty with that is just bringing them back, because
I know. Yeah, I mean,
put them in tubes and then put them with ice packs in your luggage. My luggage with one set of the spores actually got lost on the way back, and it arrived on the plane after, so that was stressful.
yeah.
And then you get stopped by customs.
Yeah, yeah.
While you're bringing live material in, so it's
yeah. Understood.
But it was fun trying, and the first step is just figuring out how to grow them in the lab. And then, yeah, there would be a ton of questions you could answer if you were able to bring back also I guess, something that represents the diversity that we're seeing down there.
Yeah, one of the reasons why I asked you about the growth too is that I was wondering if this change is kind of a continuum, if as the seaweed ages over time, or is it constantly the same way? And, just those kinds of questions.
Yeah, that was part of the reason that with the ones that we transplanted, wherever they grew enough, we did try to sample just the new growth. So I guess within a year, we did have, if the individual started off that size, it may be doubled in size.
Okay.
Sometimes, just roughly. chemists need a lot of material for extractions, so that wasn't always possible. It was always a fight between us of who was getting how much material for the different science. It was fun.
Other questions for Sabrina?
Oh, I think there's one in the chat.
Yes.
So
Yeah.
do you know if the seaweeds populations are stable throughout the year? Is there any seasonality to their growth, reproduction? That's a really good question, and something that I would absolutely love to address. Part of the problem is if someone will let me stay down there for a year or two at a time, we could look at that in a lot more detail and see what sort of the patterns are in their reproduction. So whenever we go and sample the seaweeds, we are not able to tell them apart, that they're different life stages, until they're reproductive. So if I have material that's not reproductive, I've got no idea what it is until they are. And I think only about half the ones that we collect are actually reproductive. And so far, I actually have never found a male. That doesn't mean they don't exist, but it's really hard to find them. So those kind of studies, I think, are gonna be difficult until you're actually able to be there throughout the year and can see how the populations change. That makes sense.
Alex Lowe has a question. How can you tell if they are reproductive?
Well, Alex should know that because he was on the ship with us last year. And I was usually quite excited when I found reproductive structures. So some of them have something, don't know, I guess depending on what life stage it is, because they have diploids and haploids that grow into separate individuals. So seaweeds are totally crazy. Part of what makes them so interesting. But if one part of them is reproductive, they actually have these three-pronged structures that kind of grow where their branches branch off. And that's where you can find the spores inside. And then the other one is the fertilized females grow tiny little balls on them. And then the males, I have seen pictures, but it's just a difference in the surface, on the surface that you can see, which maybe I just don't quite know what I'm looking for, but I haven't been able to actually see that, but it's really hard. And then you have to sort through, I don't know, all the samples that you collected in a dive.
So can you say something about the chemicals that you refer to?
Oh dear.
So I'm assuming that they have a bitter taste. Can I assume that?
That's a really good question. I did try it once. It didn't taste very bitter, but I also like Brussels sprouts, so maybe that's why. So the chemicals, there are what's called halogenated monoterpenes. And the thing is with those chemical defenses, it's not necessarily the taste. It can also be that they're messing somehow with the digestion of the organism, or if they get into the cells, that they're messing with the cellular processes.
Makes sense.
Hey, Sabrina. Great talk. Really enjoyed it. You're a rock star. Just wondering, so you're talking about different, well, you mentioned how some populations of the seaweeds that are more chemically similar might be a little further apart. And so I'm wondering, would you interpret that as suggesting that like seaweeds in a particular area are maybe seeded by a single event, whether it's reproduction or an iceberg scraping seaweeds from one place to a new place, and then all the seaweeds in that location then are maybe the result of that single plant and a single event and spreading out? Or
yeah,
yes, is that the interpretation there probably?
Yeah, totally. I think that is sort of my working hypothesis at the moment, is that somehow that's what's happening. And again, once I've analyzed more of my samples, we will hopefully be able to tell a lot more about that, because what you can look at with the markers that I'm using is you can look at the genetic diversity within one location, so you can see how closely related individuals are to each other by like how many alleles they share. But the other thing that you can look at is between the different sites, how much genetic exchange has been happening. So how many individuals may have traveled between sites, if that's what's happening, but in one way or another. But we can, yeah, we should be able to deduce some more of that.
So, like, especially since you mentioned difficulty finding males, I was just wondering then if we're looking at probably like some kind of clonal reproduction, which again, kind of, I think maybe supports the idea that you get one event where a plant seeds an area and then is able to spread out and produce genetically awfully similar or maybe identical organisms. So yeah, very cool.
Yeah, there's definitely, I mean, there's the sort of idea is that we don't see the fertilized females until they're actually have already grown the little ball on them. So what could have happened is that the males release their gametes to fertilize the females, and maybe then the males die off. So by the time that we actually find the females, the males have gone, or we're actually not able to see the material anymore. And the other thing is that it could definitely be some sort of clonal, some type of clonal reproduction, so asexual reproduction that's happening. Especially, it's been shown before in other areas that with seaweeds that live in sort of extreme environments, which I guess would be down there, that they tend to do more of that kind of asexual reproduction. But yeah, we'll see.
And also, how widely distributed is this species of Plocamium? Is it specific to the Antarctic Peninsula?
Good question. So because there's still so much more work to be done, not just with seaweeds in Antarctica, but anything that lives in Antarctica, we call it Plocamium cartilagineum, which is a species that is found or has been described worldwide, but we know now, looking at the genetics, that it's definitely not a different species, but no one has gone and actually described it as a different species. It does look like it's definitely all along the peninsula. There have been records from the other side of Antarctica, but I have not seen any work that has looked at whether those are actually the same species, or maybe those ones on the other side came from the other continents. There has been some work. I think they originally had about 3 different Plocamium species along the peninsula in Antarctica described because of their difference in morphology. But when you look at the genetics, they're actually all the same species. They're just growing differently. So the ones further north look a little bit different at times. And even around Palmer Station, we sometimes wondered, well, this looks really similar, but maybe it could be but turns out genetically they're the same. So a lot to do.
Any other questions for Sabrina? Help me thank Sabrina with your applause for a fantastic talk. And thank you, Sabrina, so much for spending your time with us and teaching us about the discoveries that you're making.
Thank you.
So good to see everyone, and welcome again to those of you who've joined us for the first time. I hope that you all come back. All right, thanks everybody.